Welcome back to the course Fundamentals of General Immunology, and we will continue with our module 3 and lecture number 9. So the topic of the discussion today is epitope architecture and determinants of immunogenicity. So we looked into the epitope, how these function of epitopes are modulated by types and variation in the epitopes, and also how this structure of epitopes, doses of epitopes, administration of antigens, and everything decides the immunogenicity in an individual. So we move further and we'll try to understand another important aspects of this antigen immunology, and that's antibody. So before going to antibody, let's try to understand the serum protein analysis. So when we see the protein electrophoresis of serum, we could see some pattern. Before that, let's understand what is serum. We have discussed this earlier also. So when we collect the blood into a tube with without anticoagulant, then basically the blood won't coagulate, and what we are getting is plasma actually. But when we collect the blood without any anticoagulant, so suppose you collect the blood with no anticoagulant, then after some time, if you just give a time, then the blood will clot, and this clotted mass, so the blood will clot, and we can get certain kind of fluid on the top of this clotted blood mass, and that's what we call serum. Now this serum is full of different kinds of protein which are essential for body immune response, and most of the time in immunology we study the serum proteins only, and that too particularly we are mostly talking about the immunoglobulins which are present. So when we see the protein in total, suppose whatever protein is present in the serum, we don't feel that these proteins are either positively charged or negatively charged. We consider this, that the charge of the proteins present in the serum mostly depend upon what kind of amino acid compositions are there in the protein, and therefore we can't see the size of the protein when we are, or can't predict the charge of the protein when we load it into an electrophoresis chamber. But generally the things are flowing towards the negative direction to the positive direction. Considering the proteins are negatively charged, we load all the proteins, and as I said that proteins are not always negatively charged, but when we add SDS we could overall impart a negative charge to the protein, because SDS are anionic in nature, and therefore once the protein is loaded and upon the force of electricity, once the protein has run on an electrophoretic system, then the protein migrates from negative to positive pole, and then you clearly see the segregation of protein from the serum samples. And this is very critical because you could analyze what are the different content of the protein present in the serum. In addition to this, you can also quantify the protein, like what protein percentage are more in the serum sample. We can also look into that. So once you run a gel, then one can understand that the lower migration is of gamma fraction and followed by beta, then we have alpha 2, alpha 1, and finally the albumin. But the concentration wise, albumin is most abundant protein in the serum, and followed by the other proteins as well, but most of the important content of the serum is gamma globulins, and these are very important for the immune response. So so this is a very very unique way to segregate the protein fraction from the serum sample, and most of the time we talk about these fraction only in immunoglobulins structures, because albumin is a crude serum protein which can do other functions, but mostly in immunology we are concentrate ourselves on these globulin fraction. So this is actually albumin and this is globulin fraction. So we will try to understand what is there in the globulin, and a general term which we will study here is antibody. So we will see this antibody in great details now onwards, and we'll try to understand what are the complex things which define this immunoglobulin very unique molecules, and also how this variation in the immunoglobulin can lead to variety of immunoglobulins in the human body. Say their numbers are huge, and their concentrations are also varying to different levels. And just to add up the point to the previous slide, that the concentration of all those immunoglobulins fraction or globulin fractions may increase during different disease condition. So this could be also an indication that a person might be suffering from different disease condition just based on the concentration of globulins in a serum sample. So the variation in this antibody is huge, because we every day we encounter many or different kinds of antigens which are having a very different varieties of epitopes onto them. Each epitopes are different, each epitopes is different, and based on that different epitope, antibodies generations are also different, and each antigen can bind to a very separate set of antibodies, and therefore there are huge bunch of variation exist in antibody. So if you see closely, this is a Y-shaped molecule, and there are two arms of it, and basically these are all protein chain. So these are made up of, this is a light chain and we simply call it L, and this is a heavy chain which we call H, and we could see that these two chains are bound with each other with the help of these disulfide linkages. And if we use a disulfide linkage breaker such as beta mercaptoethanol, so beta Me, then we can see that these all fractions are coming out. So we will, if we treat the immunoglobulin with beta mercaptoethanol, we could see that there are two heavy chains plus two light chains, they are coming out. So basically these immunoglobulins are a mixture of two heavy and two light chains. There are different kinds of heavy chain, and based on that we are nomenclature, we could name it the immunoglobulin based on the heavy chain. So this could be 5 different types of heavy chain. Either they could have alpha chain, beta chain, gamma chain, epsilon or delta. Based on that there are 5 different kinds of antibodies which we will discuss later. While in terms of light chain, there are 2 light chains which are reported. One is kappa and the other is lambda. So, so there are 5 different varieties of heavy chain, but there are 2 main types of light chain, kappa and lambda. So let's go back and see the structure once again. So here we have this light chain and the heavy chain. Light chain have half a portion called variable light chain, which is highly conformational in terms of its binding ability with an antigen. So the antigen could come here with this exposed area, and this epitope can bind to this particular area which we call paratope in antibody. But these are highly variable because this will change based on what kind of antigens are coming there. And similarly, we have another variable region here in heavy chain where we call it VH. So this is variable heavy chain, variable light chain. Then we have a constant light chain, C stands for constant here. And then we have the heavy chain. There are 3 domains of the constant chain. So we call it constant heavy 1, heavy 2 and constant heavy 3. So there are 3 constant region in the heavy chain. And we have this long chain of joining between the CH1 and CH2 domain, and this could vary between different kinds of immunoglobulins, and the constant region is also giving an binding affinity with complement protein, which we have discussed in the earlier classes, that these complement protein can start doing opsonization phenomena or can do a membrane attack complex phenomena to kill the pathogen. So these are integral part of the innate immune system. So these complement proteins can bind to this constant region of the immunoglobulins. So therefore these are also important. So, so basically these are considered as N-terminal domain, and these are considered as the C-terminal, carboxy terminal domain of an antibody. So let's move on and see some more details of this. So as I said earlier, and we could understand that there could be a constant region and could be a variable region. Generally this fraction is called FAB region. Generally this region is called FAB region, and in between this CH1 and CH2 domain, we have this section which can connect this CH1 CH2. This is called hinge region, and if we could look into carefully the amino acid composition of this hinge region, these are full of proline residue. So the proline is present in a large abundant quantity in this hinge region, making the flexibility to this region in immunoglobulin. So basically this can provide flexibility to the hinge region. So this is hinge region, and after that we have FC region, which stands for constant region. So this FC region is actually towards the carboxy terminal, and this FAB region is towards the antigen binding site, which is exposed to outside. And of course these are disulfide linkages which are joining the two chains of Hebbian light together. Okay, we will move further and we will try to see that there are cells which can produce immunoglobulins. We have discussed about lymphocyte, and in one type of lymphocyte we called it a B lymphocyte, which produced from bursa of Fabricius and bone marrow. So B name itself came from a bone marrow, which is a primary lymphoid organ in human being, but in case of birds this is called bursa of Fabricius. It's a name of the organ which is situated in the body of chickens and poultry, and this can provide the immune response, B cell generation to the poultry. So therefore these are innate, these are the organ of immune response in case of poultry. So this is the name, origin of B cell, origin of name bursa. So these are the two types of B cells. One is called B1 B cell and the other is called B2 B cell. And B1 B cell is actually cells which are already present in the human body. They are not say generated upon the exposure of antigen. We have looked into the earlier classes about the natural antibodies, and natural antibodies are something which are produced naturally into the system, even without an exposure of an antigen, such as the natural IgM molecules. So we can look into this immunoglobulin having M chain little later, and there is no memory and no somatic hypermutation, which is also a term which we'll discuss in the next class about how this rearrangement of the gene in the human body or the cell of the immune system can decide the variation of immunoglobulin among the individuals. And this one is called B2, which is producing a different types of immunoglobulin, such as immunoglobulin G, immunoglobulin A, immunoglobulin E, and of course immunoglobulin M. And these are a part of adaptive immune system which are produced upon the exposure of antigen later in the lifetime or life cycle of an individual. These are not present right from the birth, and they are acquired in terms of its generation. They carry the memory. They do have a process called somatic hypermutation that could lead to variation of antibodies, and also they have a random specificity. So they can have a very generalized way of specificity that can bind to some very unique pattern of immunoglobulins. Right. So we'll move and we'll see a different kind of isotypes of antibody. So when we call isotypes of antibody, these are the antibodies which are or different kinds of antibody which are present in serum. So the isotypes are based on the types of heavy chain they carry. So they can carry either M type of heavy chain, G type or gamma type of heavy chain, alpha type of heavy chain, epsilon type of heavy chain, or delta type of heavy chain. So accordingly they can call IgM, IgG, IgA, IgE and IgD. So there are 5 different kinds of isotypes. If we see closely, IgG, IgE and IgD are monomeric in nature. So they have only single immunoglobulin molecules carrying 2 heavy and 2 light chains. But IgA and IgM are multimeric protein. So IgA is a dimeric protein which are joined with each other with this J chain, and this M is actually a pentameric protein which is having a 5 unit of immunoglobulins to make it as a highly specific or most highest affinity antibody, because they have 10 pockets to catch the antigens onto the surface, while IgG have 4 in their surface, so they can catch 4 molecules of antigens into their surface. So based on that, and each of these types are having some specific function. They have some very specific cell type that produces it. They have a typical pattern of their function, and their clinical relevance are also very important. So presence of this immunoglobulin in the body system can define a lot of things. So for example, IgM is actually produced by a B1 cell and the naive B cell. So there are 2 kinds of immunoglobulins which are very important, IgM and IgG. That's IgG is the most abundant immunoglobulins in the human body. So the very first antibody that comes into any infection is IgM. Later on there is a class switching phenomena, and that could lead to conversion of IgM into IgG. So that we could look into our further discussion on somatic hypermutation, how that happened. While IgG is actually produced by memory B cell, that means if the cells are exposed to the antigen, they understand that what kind of immune response that's needed to remove the antigen, and therefore you always see that there is a response of memory involved in generation of IgG molecules. IgA is also a plasma cell specimen. These are actually secretory immunoglobulins. So they can secrete. So whenever there is a secretion, such as the nostril secretion, the lacrimal secretion from the eyes, or say saliva, they contain these secretory immunoglobulins, IgA. These are very local in nature. IgE, in the other way, is actually the immunoglobulins which are mostly produced upon allergic infection or allergic reaction, also at the time of some parasitic infection. So IgE, presence of IgE immunoglobulins is an indication that the person might have an allergic reaction or say some sort of parasitic infection going on. So IgE is some indirect indication of allergic inflammation. IgD, on the other hand, is actually present on the surface. These are not freely available. So these are specifically bound to the cell membrane. So they are not solubilized. They are not freely circulating in the blood circulation, and therefore they are mainly surface bound and not secreted out. If we see the function of it, we could see that IgM is actually responsible for complement activation. We have looked into the different pathway of complement activation, and one pathway was antigen antibody complex. So antigen antibody complex can lead to complement activation that could lead to the microbial destruction. IgG is responsible for antibody dependent cellular cytotoxicity, ADCC, or opsonization. Opsonization is a phenomena where a complement protein can bind to very specifically on the pathogen in order to help them on phagocytosis. So they can allow the engulfment of the specific pathogen by the phagocytic cell. So that's what called opsonization. IgA is actually neutralization. Neutralization stands for they can simply bind to the stuff so that the antigens or any kind of foreign molecules are not available to the body system to do an effect. So this is what the meaning of neutralization. So one can neutralize the thing. So IgA function on neutralization. IgE, as I told, this is mostly involved in activation of allergic reaction, and allergic reactions are mostly done by degranulation of the mast cell. So mast cell, once activated, starts secreting histamines, and these histamines can start doing a lot of other function, could lead to allergic infection. And IgD is simply some sort of regulatory function in the body. The site of IgM, mostly these are all present in blood secreted in different parts of the body, and some of them are secretory, so they can present on the mucosal surface. For example, IgA is present on mucosal surface. The saliva, the nostril as the nasal secretion, the lacrimal secretion, any secretion of the body mostly contains this IgA molecule, which are secretory in nature. And remember, these are dimeric in structure because of this J chain, while IgM is pentameric in nature. Ig is again related to allergic infection, so these could present in skin and mucosa, and IgT is mostly present on upper respiratory tract or tonsils. Clinically, they are all relevant, and their presence and excess amount in the body system could alarm the body towards some infection or some kind of processes which are happening in the body. For example, IgM, as I told, this is the first antibody that comes into the picture. So this is a very specific antibody that can decide the early infection. Early infections are also called as acute infection. So they can give an indication that the person is recently started suffering from some infection, because IgM response will increase, while IgG is actually a long-term immunity, because their memory response are involved, and therefore once a person is suffering from a disease condition, the antibody which comes into the circulation for longer duration of time is mostly IgG types, and therefore these are indication of chronic infection. Chronic means long lasting infection, the infection which lasts for a little longer duration of time. So IgG is responsible for those, indicating chronic infection. While the mucosal surface, mostly the gut epithelial lining, the lung, because lung is full of surfactant, lung is full of the viscous material, mucus secretion, because the pool passage is moist in nature, and they carry this mucosal secretory antibody into them. Also lung epithelium, they also the gut epithelium, which is full of mucus, can have a secretory antibody. So both lung and gut, they have high amount of IgA specific antibodies. And IgE is especially related to allergic infection. So asthma is one such condition where we could see that IgE production is increased in the human body, and D is mostly, it's not in the secreted form, so we can't see that much function with IgD, but mostly these are associated with the surface and they are bound with the immune cells. So we'll move further and see the different subclasses of. So as I told, there are 5 classes based on the heavy chain: IgM, IgA, IgG, IgA, IgE, and IgD. IgG is further divided into multiple subclasses, and if you can look into that, so IgG is actually a monomeric form. So one can easily see that we could say IgG1, IgG2, G3 and G4. There are very subtle deviation into their structures which are present. We could see that the hinge region is quite large in case of IgG3 molecules, and this could lead to deviation in the function of these IgG3 molecule, while IgG1, G2 and G4 looks pretty similar in their structure, but they have a variation in amino acid sequences. As I told earlier also, IgG is the most abundant immunoglobulin present in the serum, and out of that IgG1 is the most abundant, around 70% in the serum. The concentration of IgG is pretty high. Then followed by IgG2 and G3, and lastly G4. The concentration is very less in the human body. Their functions are also very different, although these are related to chronic infection. This is an indicator of chronic infection. They have a very strong opsonization activity or neutralization activity. They can also do ADCC, which is antibody dependent cellular cytotoxicity. So basically antibody can bind to the cell and start doing some sort of cytotoxicity just to remove or nullify the effect of external harmful substances. IgG2 is weak in opsonization activity, and they generally respond towards a polysaccharide. So their response are mostly abundant when the antigens are having polysaccharide nature. Again, IgG3 is having a very strong complement activity. People have reported to have this antibody upon viral infection, so they are considering as an antiviral. And G4 is mostly anti-inflammatory, so they can work against inflammation, and also they have a poor effector function, so they can't do very high quality of neutralization or opsonization. So in terms of complement activation and binding to Fc gamma receptor, so they are strong, G1, while G2 is weak and G3 is again strong and weak. These are some of the parameters which people have, these are some of the parameters which one has to remember looking into the different function of subclasses of IgG molecules. Clinically as relevant, most effective against protein antigen such as viruses and toxins, G1, while G2 is mostly responsible for polysaccharide, specific polysaccharide which are present in bacterial cell wall. Most of the time the polysaccharide concentration is very high in capsule, so we called it a smooth, where the capsule is actually highly pathogenic in nature, while in another case we called it rough, where the capsules are not that much immunogenic. So capsular polysaccharide is again a very important antigens against which the body immune response are produced, and particularly we could look that IgG response are coming, but there is another subclass of IgG, that's IgG2, and the G3 is again important for a viral infection, and they have a very short life. So these immunoglobulins are, of course, these are there is a specific lifespan of these immunoglobulins. Some are short, some are very long lasting immunoglobulins. So these are particularly very short lived, and G4 are involved in chronic exposure, and to some extent when there is a chronic allergy condition, then we could see that there is some sort of response towards G4 in addition to IgE molecule, because allergic and some sort of mast cell related biology mostly towards the IgE response. So we could see that these functions are quite different to these immunoglobulins subclasses of IgG. Alright, so we will see this antibody isotypes and what are the actual function in terms of a small tabulated form. So here we can say that minus stands for does not apply, plus double plus and triple plus depends upon whether it is apply or strongly apply, moderately apply and all. And there are 5 parameters which we could understand. One is called neutralization. Neutralization stands for once there is a entry of an antigen, the antibody can bind and simply won't allow this antigen to go outside or to infect or alter any other cellular processes, and therefore this is actually neutralized right in the entry point, and therefore this is called neutralization process. Mostly we talk neutralization in terms of viruses, so we can say that virus neutralization in terms of immunoglobulin. Opsonization is again we have discussed it multiple times when when the bacteria can be phagocytized by macrophages with the help of antibody or complement proteins. So, the opsonization is basically a phenomena of eating a pathogen with the help of antibody and complement. So, that is also important. Sensitization for killing by NK cell. NK cell is natural killer cell. So, we have understood in the cellular system, earlier we looked into the innate cells. There are cells in the body which have a capacity to kill by their own way. Therefore, they are called natural killer cell. They are least abundant in the cell. They can come and activate a direct killing of the pathogen. So, they are the cells of the innate immune system. Their numbers are very less in the human body. Sensitization of the mast cell, the fourth one, where the allergic infections are involved. The mast cell will start degranulation and produce a lot of chemicals, and one such important chemical is histamine. And then of course, complement system activation is the last one. So, these are all 5 important processes, and we could look into what are the different immunoglobulins which are key on that. Definitely, if it comes to sensitization, we can directly concentrate our discussion towards IgE. When it comes to opsonization, we can see that this is mostly done by IgG1 molecule. So, we will just highlight something which is strongly there. For complement activation, we can see that IgM is actually very important, and also IgG3, that is what we discussed in the earlier slides. And in terms of neutralization, we do not see there very specifically any player that does neutralization very strongly, but every immunoglobulins, presence of immunoglobulins could do a neutralization to some extent. IgM, IgA, IgG1, G2, G3 and G4. So, all the subtypes of the IgG molecule can do neutralization, while NK cell activations are mostly done by IgG subtypes, and particularly G1 and G2. So, what we understood here is these immunoglobulins are very important. There are different types of immunoglobulin, their functions are different, their site of origins are different, their variation will depend upon how these are acquainted, how these are identifying the pathogen. There is internal gene editing phenomena that is going on in the B cell that could lead to variation in the immunoglobulin. So, these immunoglobulin can do variety of function. Some of them are listed here, and gradually when we will go across the course, we will try to learn little more complex biology of the antigen antibody reaction, and how this beautiful interaction of antigen antibody could lead to protein-protein interaction biology very relevant to the body system, just to maintain the homeostasis in a proper way. So, we will move on to further discussion on the same topic of antigen antibody interactions in our next class. Thank you.